Great. Thanks, Levi, and thanks to all the organizers, and thanks to everyone who's watching. Really amazing effort. Really love the idea of this Microbiome Virtual International Forum, and I'll join myself for sure and encourage others to do so. So what I wanted to do today was to share a little bit on how we might be able to target the microbiome to promote health and end cancer. And notably, I do have a few disclosures. And so before I get started into the talk, really wanted to thank everyone who's contributed to this work. First and foremost, patients and their families, all care providers, the Melanoma Moonshot team and research teams, and our microbiome program at MD Anderson, the leadership, supporters, and collaborators worldwide. And really thank you to everyone worldwide for your contributions to advance care through science. And it's really critically important that we continue to do this. And so I think we can all agree that we've made major advances in cancer treatment with the use of immunotherapy and other approaches. Immunotherapy has made its way as one of the very pillars of cancer care, and we've actually seen an overall decline in cancer-related mortality for the first time. But what I would also argue is that there's still a critical need to improve responses to cancer treatment and to limit toxicity in patients with established cancer, as well as opportunities to prevent cancer altogether. And through translational research, we've identified a number of factors that impact tumor growth and response to cancer treatment that can be targeted. And we summarized these findings. Gulnaz Murad, who's a postdoctoral fellow in my group, published these findings in Cell just a few years ago. And this includes factors within the host, like things within the tumor and tumor microenvironment, the host genomics and epigenomics, systemic immunity, systemic factors like sex hormones, and also the microbes that are within us. But it also includes factors that are extrinsic to the host, things like environmental exposures, psychosocial factors like stress, lifestyle factors like diet. We'll talk a little bit more about this, and the microbes that surround us. And so considering that humans and organisms represent a complex ecosystem, what is the role of microbes that coexist with us on cancer and on therapy response? And so interesting little piece of trivia. So 3.5 billion years ago, microbes actually helped to shape the earth for future forms of life. They actually gave us the very oxygen that we breathe today. And 3.5 billion years later, it's really clear that microbes are pervasive in our environment and also within living organisms. So take a human being, they have around 20,000 human genes, which represents only 1% of the total genomic content. But within that same individual, there's between 2 and 20 million microbial genes, which represents 99% of the total genomic content. And importantly, these are inherently modifiable. And advances in next generation sequencing have really allowed us to better understand these microbes and really their impact on our own physiology. And the importance of the microbiome in health and disease is increasingly evident, and this includes in cancer, where microbes within the tumor and surrounding tissues, as well as microbes within the gut and other niches, can actually impact whether or not cancers develop, how they respond to treatment, and other physiologic processes. And Doug Hanahan recently added polymorphic microbes to his very hallmarks and enabling characteristics of cancer, as updated and published in Cancer Discovery. And so I really posed the question, could microbiome targeting become the next pillar of cancer care, right next to immunotherapy there, with strategies to monitor and modulate the microbiome to treat, intercept, and maybe even prevent cancer altogether? And so some people ask me, well, Jen, how'd you get started in studying the microbiome? Well, we first studied the role of the intratumoral microbiome in cancer, but we did it somewhat serendipitously. And so, as Levi mentioned, I started off my career at Massachusetts General Hospital and Harvard University, and there we used a model system to study stromal-mediated resistance to treatment in melanoma, really posing the question, tumors aren't just balls of cancer cells, and components of the tumor microenvironment alongside tumor and immune cells actually mediate therapeutic resistance. And so to study this, we developed this 3D culture system where we co-cultured cancer cell lines along with stromal cells and along with drugs, in this case, melanoma cell lines and BRAF inhibitors. And what we found when we did this is that there were certain stromal cell lines that were actually able to confer therapeutic resistance to treatment with BRAF inhibitors and very reproducible, and found that these stromal cell lines were actually secreting hepatocyte growth factor, or HGF. We actually turned back to our patient samples and found that if patients had HGF within their tumor microenvironment, that they were actually resistant to treatment, and actually published these findings in Nature over a decade ago. And really, following this, we wanted to use that same model system to study stromal-mediated resistance in other cancers, and here's where the twist came in. And so, again, at the time, I was not only treating patients with melanoma, but also with pancreatic cancer, and worked with Ravid Straussman and Todd Golub at MIT to use this same model system to study resistance to chemotherapy in colorectal cancer and pancreatic cancer. And so in this case, we actually took the same 3D culture system and studied cancer cell lines, in this case, either pancreatic cancer or colorectal cancer, along with stromal cells and along with drugs, in this case, chemo. And what we found when we did this is that there was actually this one cell line, this HDF, or human dermal fibroblast line, that was actually consistently able to confer resistance to treatment with chemotherapy across multiple pancreatic cancer and colorectal cancer cell lines treated. We got really excited about this. We were very excited and very reproducible, and really scoured human tissues to come up with potential enzymes, which we thought it was. But then, interestingly, when we were testing the cell line in the laboratory on routine testing, it was actually found to be contaminated with mycoplasma, which, you know, most of us would just groan and kind of give up. But Ravid is brilliant, because they said, well, wait a second, what if this mycoplasma is actually responsible for the therapeutic resistance? And so what he found is when he actually eradicated the mycoplasma, there was no longer rescue. When he infected another cell line, it conferred resistance, did whole genome shotgun sequencing of the HDF preconditioned media, and lo and behold, found mycoplasma, and found that these bacteria were actually breaking down gemcitabine into its inactive form. And so Ravid and Todd, came over and saw us and actually asked for our help, and we were actually able to validate these findings in human samples. So when we took patients to the operating room, resected their tumors, if we brought them back to the laboratory and plated them out on agar plates, we actually found that they were chock full of bacteria. Whereas if we did surgery for non-cancer indications, such as pancreatitis, we actually found that they were devoid of bacteria. Actually sequenced these bacteria, found that these were largely gamma proteobacteria, along with Curtis Huttenhauer, visualized these bacteria in tumors, and then took it back to a mouse model where we co-targeted these intratumoral microbes by treating with an antibiotic, ciprofloxacin, along with treatment with chemotherapy, and cured all the mice of their cancer, and published these findings in Science, just about 5 or 6 years ago. And so that's how I really got started studying the microbiome, was through this collaboration with Ravid Straussman, and really have continued this work as well. And so now we know that microbial signatures are now being identified across all tumor types, with opportunities to target these to improve outcomes and maybe even prevent cancer altogether. And so Ravid has continued his brilliant work. He's now at the Weizmann Institute, and has shown that intratumoral bacteria can be identified across all tumor types, though the composition differs depending on the histology. In addition to this, other investigators like Rob Knight have mined TCGA data and actually identified microbial signatures in tumors and even in blood of patients with cancer. And Yardenia Samuels actually worked with us and showed that microbes can actually contribute to anti-tumor immunity by recognition of microbial neoantigens that can actually be processed, presented, and recognized by antigen-specific T cells. And I posed the question, could we prevent cancer altogether? And I think the answer could be yes. By mining these and looking for signatures within these tumors, could we identify, microbes that we could actually target? And there's a precedent for this. Think about HPV. Now, I will say that there's been some scrutiny of Rob Knight's data and showing some potential errors in the processing, which led to some calling this into question. Nonetheless, I think we can all agree that there are microbes within the tumors, although I think we need new standards and approaches to really be able to make sure that to really minimize the risk of contamination and really separate signal from noise. And so we and others are doing just that. We have this program for innovative microbiome and translational research at MD Anderson, Prime TR, where we have a small army of individuals who are really dedicated to just this. And we're not only mining data that's been collected for other reasons, for microbial signatures within tumors, finding some really interesting hits. But we're also doing careful prospective collection of tissues and doing microbiome profiling within those carefully curated samples. Also using novel technologies such as spatial transcriptomics and single-cell sequencing to identify microbes within tumors and to really validate those. And we've really come across some very interesting findings. Gulnaz Murad, who published that review in Cell, also has done some great work with the Moonshot team studying GBM, and has shown that in both brain metastases and glioma, you can actually identify microbes within these tumors that actually may even be seeded from the mouth, which is really interesting, and has looked at potential interactions with the immune cells as well as with the tumor cells. And stay tuned for these findings, which hopefully will be published within the next few months. And studies using spatial transcriptomics actually have revealed unique contributions of intratumoral microbes to promote cancer cell invasion and modulation of the tumor microenvironment. Susan Bowman has done some beautiful work and really showed that cancer cells that are infected with bacteria invade their surrounding environment as single cells and can actually recruit myeloid cells that suggest that their microbiota and tumors are actually organized into microniches, which promotes progression. And she had a great paper published in Nature not too long ago on this. And so that's the tumor microbiome. But in addition to microbes within tumors, we know that microbes within the gut can profoundly impact immunity and immunotherapy response. And if you think about it, all along this long alimentary tract, on one side, there are a dense network of microbes. And on the other side, there's this dense network of infiltrating immune cells. And there's this constant to and fro and dynamic interaction and, candidly, education of the immune system by those microbes and by their byproducts. And early studies really supported a role for gut microbes in shaping response to therapy, with validation in numerous clinical cohorts later on in patients with immune checkpoint blockade. So some of the earliest work came out of Memorial Sloan Kettering, where investigators like Eric Paymer and Ying Tower showed that in patients with leukemia who were being treated with stem cell transplant, if they had a more diverse gut microbiome, which is generally associated with better overall gut health, that they had much better survival. And then in preclinical studies, Tom Gajewski and Laurent Sitvogel showed that depending on what the bugs look like in the gut of mice, dictated whether or not they responded to treatment with immune checkpoint blockade for cancer, and furthermore showed that you could actually change the microbiome and make those mice respond better. And so we were really inspired by that work, by that early work, and studied a cohort of melanoma patients who were being treated with immune checkpoint blockade, primarily with anti-PD-1. We sequenced their oral and gut microbiome longitudinally and also looked at the tumor and immune cells. And what we showed is that patients who responded to treatment with immune checkpoint blockade first had a more diverse gut microbiome, much like those had seen with stem cell transplant, and also had different bugs in their gut. And so they had a higher abundance of things like Ruminococcacea, Fecalibacteria, and Clostridiales, where patients who failed to respond to treatment had a higher abundance of things like Bacteroideales. And we co-published these findings back-to-back in Science with Tom Gajewski and Laurent Sitvogel, who also had studied human cohorts in patients with melanoma, lung cancer, and kidney cancer. And so this brings up a lot of questions, right? First of all, can we actually use the gut microbiome as a biomarker of response to immunotherapy? And I can tell you that though gut microbial signatures are associated with response to checkpoint blockade, when people first looked at this data, if you look at those three separate cohorts, there was a lot of variability and not really a unifying signature as to which bugs were associated with response. However, nonetheless, now that as we've really done more profiling and more patients, we've come to recognize that there may be some unifying signatures, and gut microbes are actually strongly associated with response and toxicity to other forms of treatment like CAR T therapy, with patients who receive antibiotics demonstrating shorter survival and higher toxicity, and specific taxa that are associated with prolonged survival and lower toxicity. And some of the top bugs here are some of our favorite bugs like Ruminococcacea, Fecalibacteria. And in fact, two recently published papers in Nature Medicine suggest that it's not only the good bugs that matter, but it may actually be not having the wrong bugs in your gut that actually makes a big difference, really suggesting that it's not only the presence of a good microbiome that matters, but the absence of deleterious taxa and their functional consequences that are associated with a good response and lower toxicity to treatment. And I really go back to this paper from Yasmin Belkade published in Cell in 2014, which really kind of calls out the importance of these gut microbes and microbes in general in really kind of homeostasis and when you have dysbiosis in really promoting inflammation. And so another big question is, can we actually modulate the gut microbiome to enhance response to immunotherapy and to abrogate toxicity? And I used to just say this optimistically, but now there's actually hard data that this actually can be true. And so efforts to target gut microbes to improve response using fecal microbiota transplant can be effective. And so just a few years after we published those back-to-back papers in Science, there were two clinical trials published in Science demonstrating that treatment with fecal microbiota transplant, or FMT, can actually overcome resistance to immunotherapy in patients with metastatic melanoma. So Rez Baruch and Duakar Davar published back-to-back papers in Science showing that FMT could actually promote response in immunotherapy refractory melanoma patients, and showed not only do you get a decent response rate in these patients, but you can also see changes in the microbiome and see differential taxa, including Clostridioides and other favorable taxa that are enriched in responders like Ruminococcacea. In addition to this, you see a night and day difference in serum metabolites, which is really interesting. And in fact, does it even take a complete responder donor? Well, it doesn't necessarily. And a recent clinical trial published in Nature Medicine showed that treatment with FMT from healthy donors alongside treatment with anti-PD-1 in treatment-naive patients with metastatic melanoma was associated with a high response rate. And really nice work from Bertrand Rowdy and Saman Malecki. Note that this was a single-arm study, so there wasn't a comparator arm. Nonetheless, I think, compared to historic controls, a pretty high response rate for these treatment-naive patients going on to anti-PD-1. And so kudos to that team. Nonetheless, optimal FMT donors really remain undefined, and opportunities exist to engineer next generation synthetic communities to improve response and overall immunity. And so to help address this, we've actually studied cohorts of potential FMT donors, including complete responder donors and healthy donors, compared to non-responders. And we've identified biomarkers to confer response that really transgressed across both healthy donor and complete responder donor cohorts. But when we go back to the mouse models, what you'll find is that it's not all healthy donors that actually confer therapeutic benefit. And it's not even all complete responder donors. And so, there's still a lot of work to be done to really demonstrate which is that optimal donor. And I think we can get there. In addition to this, we're working with others, including Michael Overman at MD Anderson and Michael Fischbach, as well as Canvas Bio, to engineer designer microbial consortia to enhance response to immune checkpoint blockade, and actually have now engineered a synthetic community of microbes that we're testing in preclinical models, performs just as well as a whole FMT, and hoping to roll that out into clinical trials within the next year or two. And another question is, how about probiotics? And data on the use of probiotic formulations and limited consortia to improve response have been mixed, with some demonstrating efficacy while others do not. So a really nice study that was published in Nature Medicine from Monty Powell and Nasli Disman showed that treatment with a live bacterial product, the CBM 588, which is a commonly used probiotic in Japan, in combination with CTLA-4 and PD-1 blockade, immune checkpoint blockade, was effective in treating patients with metastatic kidney cancer. Really showed much better response when compared to the CTLA-4 and PD-1 blockade alone, both with overall survival as well as with relapse-free survival. However, not all probiotics are necessarily good. We actually, in a science paper published in just 2 or 3 years ago, we showed that in our melanoma patients, up to 30% of our patients reported taking over-the-counter probiotics. And when we looked at their survival, they did numerically worse, although this did not reach statistical significance due to the limited size. Nonetheless, we brought this back to a preclinical model and actually gave germ-free mice a rock star microbiome, if you will, by giving them a complete responder donor FMT, then treated them with one of two probiotics we bought off the shelf, either a probiotic that contained bifidobacterium versus a probiotic containing lactobacillus. And we showed that after implanting tumors and treating with checkpoint blockade, if those mice received either of the probiotics, they actually had worse outcomes with accelerated tumor growth and poor response to checkpoint blockade, and they actually had impaired immunity. We also worked with Seres Therapeutics to test a consortia of microbes, a SEER401, in a randomized placebo-controlled trial, and gained some really interesting insights into the many nuances of this therapy, really kind of showing that the vancomycin preconditioning regimen potentially deleteriously impacted the gut microbiome prior to treatment with this limited consortia of microbes. So I think there's a lot of considerations that we need to take into account. And one interesting question is, how do these microbes actually help shape response? And targeted microbial intervention in the gut or tumor can actually improve anti-tumor immunity through these induction of these tertiary lymphoid structures and B cells. And so B cells and tertiary lymphoid structures are actually associated with improved anti-tumor immunity and response to immunotherapy. And we published back-to-back papers in Nature a few years ago, really showing that these B cells and TLS are signatures of an improved response. But at the time, we really didn't look at the relationship to the gut microbiome. Nonetheless, others have, and we're starting to do this as well. And what others have shown, and this is beautiful work by Abby Overaker-Delgoff, is that by giving microbes into the gut, you can actually induce these tertiary lymphoid structures in tumors and improve immunity and potentially immunotherapy response. And there are really numerous opportunities to engineer bugs as drugs to treat cancer and other diseases. Michael Fischbach had a great paper in Science not too long ago showing that antigen-engineered commensal bacteria, Staphylococcus, can actually improve response to immune checkpoint blockade via topical administration in preclinical models. And we're hoping to work with Michael to actually bring this forward to preclinical or to clinical trials, actually. And though approaches to use microbes to treat cancer have been used for many years, there's unique opportunities to leverage novel approaches to use bugs as drugs in next generation therapeutics. And so I think real big opportunities now and real initiatives. Now, how about the role of diet? And so given the critical role of the gut microbiome, what's the role of diet and other factors in response to cancer treatment? And so we know that microbes in the gut can be influenced by a number of features, including diet, antibiotics, and environmental factors. In fact, antibiotics have really been highlighted in the setting of treatment with immune checkpoint blockade, where numerous studies have shown that patients who receive antibiotics before treatment with immune checkpoint blockade for cancer have worse outcomes, markedly worse response rates, markedly worse survival. But not all antibiotics are bad, and some targeted antibiotic approaches can actually improve anti-tumor immunity. And Susan Bowman published some really nice findings, about 5 or 6 years ago, showing that treatment with metronidazole can actually have anti-tumor effects in certain clinical models and preclinical models. And these results have potential implications far beyond cancer. And so in our patients, we actually studied the role of diet in response to immune checkpoint blockade, and showed that if patients report sufficient dietary fiber intake of at least 20 grams per day, they actually had much better outcomes to treatment with immune checkpoint blockade. They were 5 times more likely to respond to the cancer treatment. And took this back to a preclinical model with John McCulloch and with Giorgio Trinchieri at the NCI, showed that if you give mice a low-fiber diet, they fail to respond to treatment with anti-PD-1 and fail to activate T-cell signaling pathways. And this is galvanized by data from Romina Goldsmid, who actually showed that gut microbes and dietary fiber can actually shape the tumor microenvironment by monocyte reprogramming. And so the fiber matters. And so we're now testing fiber and diet as a precision intervention in cancer, where we hypothesize that a whole foods-based fiber-rich diet will actually modulate the microbiome and enhance systemic and anti-tumor immunity. And this is the Diet and Immune Effects trial. We actually have a bionutrition research core at MD Anderson, where we actually have chefs that will make meals and prepare them, package them, and ship them anywhere in the world. And it's actually supported by the CRAV Foundation. So thank you to the CRAV Foundation, among others. And we're seeing success with this. This is a patient that Jen McQuaid and I treated, who came in, had metastatic melanoma, several sites of disease, was randomized to receive one of these dietary interventions alongside treatment with immune checkpoint blockade and actually had complete regression of her tumors. And she's alive and well and doing just fine. And what we find from these studies is that the dietary intervention is tolerable, excellent compliance. You can get people up to 50 grams a day, but as soon as you take that diet away, they go back to their old dietary patterns. We do see an increased interconnectedness of the gut microbiome and dynamics after really we institute this intervention. And we see really interesting changes in serum metabolites as well. And this is great work from Jen McQuaid. Now, what about the use of antibiotics? So as a surgeon, I can tell you that we know that antibiotics can disrupt the gut microbiome. And so we studied the use of perioperative antibiotics in patients with early stage resectable melanoma. Again, we know that studies have shown that patients who go on to checkpoint blockade, if they get antibiotics, they fail to respond to treatment. We know that antibiotics can disrupt response to vaccines in humans. And in patients with cancer, we routinely give perioperative antibiotics in many cases, even in settings where the risk of surgical site infection is only 1% to 2%, and despite unclear benefits of perioperative antibiotics. And so we ask the question, are we doing harm? And we hypothesize that a single dose of perioperative antibiotics would actually negatively impact the gut microbiome in terms of taxonomy and function. And so what we found when we did these studies is that treatment with a single dose of perioperative Ancef was actually associated with substantial disruption of the gut microbiome out to day 90 with defects in gut microbes, immunity, and the metabolome. We actually saw an increased frequency of exhausted CD8 T cells and a reduction in interferon gamma in the periphery. And we are also studying the use of targeted antibiotic approaches to favorably shape the gut microbiome and immunotherapy response. And what we found is that if we use a targeted antibiotic approach, we can actually improve response to immune checkpoint blockade. And again, a shout out to Susan Bowman. This was with treatment with metronidazole. What we found in a retrospective cohort is that it parallels these findings when we studied patients who had received metronidazole before treatment with immune checkpoint blockade. They actually had better outcomes. And we saw favorable shifts in the microbiome in mice that were treated with metronidazole, with a reduction in the deleterious taxa and a bloom of favorable bugs. And so again, I pose the question, could microbiome targeting become the next pillar of cancer care with strategies to monitor and modulate the microbiome to treat, intercept, and maybe even prevent cancer altogether? From a profiling standpoint, I think, we're getting there, right? Jack Gilbert I saw was on, and people are developing these smart toilets that can actually, you sit down, it reads your fingerprint, and then it profiles your microbiome real time, can actually send back data to your smartphone, tell you how good is your microbiome and give you that feedback. There are also 3D printed pills that you can swallow. It'll sample your microbiome all along its length. There's even breathalyzers for disease, and Alstone has this breathalyzer that measures volatile organic compounds. In addition to this, from an intervention standpoint, diet, microbiota interactions, and personalized nutrition is certainly here. There have been great proponents of this, and your microbiome could even play a role in your COVID-19 response. And so how about the next frontier? And we know that cancer and other diseases are occurring in people at younger ages with opportunities for interception and prevention. And could exposure to antibiotics and other factors that disrupt the microbiome be contributing to higher rates of disease and cancer? And can we target the microbiome in cancer prevention? And think about, all along the past few decades, if you think about, we instituted the use of antibiotics to treat infectious diseases, and sure, you see a reduction in all these infectious diseases, but you also see a concurrent increase in inflammatory diseases. And could the overuse of antibiotics be fueling our modern plagues? Really a quote from Marty Blazer in his Missing Microbes book. And, this could have real credence. And, a great paper from Tim Rebick and Shuji Aguno out of Harvard asked the question, is early onset cancer an emerging global epidemic with current evidence and future implications, and implicating things like the microbiome in really driving early cancers, along with obesity, Western style diet, sleep patterns. Not only do they highlight these issues as drivers, but they also offer tangible solutions to really tackling this, not only from really the grassroots effort, but also public health authorities and policymakers to promote healthy lifestyles from childhood? Can we actually change our school lunches and favorably impact our microbiome and health overall? And, I really pose the question, could we work together to better understand genetic susceptibilities, the microbiome, and numerous other factors that contribute to cancer and other diseases? And when you think about it, the same factors that are influencing cancer are also influencing other diseases. What tops cancer with regard to mortality? Heart disease, right? And, the microbiome can actually impact that as well. And can we actually monitor and modulate gut microbes, diet, and other variables in cancer treatment, and also to promote overall health? And so I think, really to summarize, I think we can help address these issues on a global scale and also as individuals, and the choices we make every day have a tremendous impact on our microbiome and also on our physiology and also on our planet. And if you think about it, a lot of us have been living in this left side of the screen where, meat eating diet, a lot of inflammation, a dying planet. Can we shift our focus and go to a more plant-based diet, healthier lifestyles, really help ourselves and also help our planet in the meantime as well. And so with that, I really want to conclude the talk, thank patients and their families, really thank MVIF organizers and participants, thank all of you for your attention and to the huge numbers of people who contributed to all this work, and really thank you for the opportunity to speak here today.